I'd like to go through a few questions that people tended to get wrong on the digestive homework. The vessels into and out of the liver was one area that students needed a little more review. The hepatic artery brings oxygenated blood into the liver. This blood also contains waste that's picked up from other regions of the body, so it needs to be cleaned. The hepatic portal vein also brings blood into the liver. This blood has already been to the tissues within the abdominal cavity, such as the intestines, stomach, spleen, so it's no longer oxygenated, but it does contain waste elements that were picked up from these regions. So this blood definitely needs to be cleaned. So the liver has two incoming vessels bringing blood to it for waste removal and cleaning. Once the waste has been removed from the blood, it is now ready to return to the general circulation. The hepatic vein brings this freshly clean blood away from the liver. Specifically, it brings the blood to the inferior vena cava, then it will go to the heart next. Finally, the waste that was removed from the hepatic portal vein and hepatic artery is taken away from the liver via the bile and stored in the gallbladder shown in green, and until it can be dumped into the GI tract and removed from the body via the feces. Let's go through this again while also viewing the microscopic view of the sinusoids within the liver. On the right, we see a portion of a liver lobule. The hepatic artery is in red. This blood enters the liver with oxygen and waste. The hepatic portal vein is in purple. This blood also enters the liver with waste from the abdominal cavity. Both of these vessels bring blood into the liver. In the sinusoid, they are on the outside and send blood toward the center so that the cells can remove their waste. The waste goes the opposite direction from the cells out to the edge of the sinusoid into the bile duct in green. The bile ducts from all over the liver drain their waste-filled bile to be stored in the gallbladder outside of the liver. The hepatic vein is in blue. This is where the clean blood leaves the liver. In the sinusoid, the central vein gets the clean blood. All the central veins throughout the liver bring their clean blood to the hepatic vein, and this is the blood that goes back to the general circulation. The hepatic artery, hepatic portal vein, and bile duct together form the portal triad. On the histology portion, simple columnar epithelial tissue, as we see here on 3 separate villi, are what line the small intestine. They're 1 cell layer thick, so we can allow for food to be absorbed, but it is a very long cell, so it also offers a little bit of protection. So simple columnar epithelial tissue lines the small intestine. The mucosa, in this image we see the esophagus, but this pattern holds true across the entire GI tract. It has both muscular and epithelial tissue. So yes, of course, epithelial tissue will always be in the mucosa, but muscular tissue, remember we have this muscularis mucosa. You can see that here, but as a little thin line here. So from this thin line here, all the way up into this epithelial tissue, this is all mucosa. So where we can find muscular tissue, we can find muscular tissue in the muscularis externa, but the only place that we can have muscular and epithelial tissue is going to be the mucosa. On the large intestine, most of you guys got correct all of the regions of the large intestine. The 2 areas that people seem to struggle with were Taenia coli. That is this distinctive ribbon, single ribbon of muscle. Now haustra are these pouches that we see that make the large intestine a very, very distinctive. So there are these pouches that are all along the entire length of the large intestine. Many of you answered diverticula. I'm assuming you just Googled pouches in the large intestine and just got that and didn't actually listen to the lecture. So nowhere in the lecture did we ever talk about diverticula. That is because diverticula is actually a pathology that we did not discuss. So I'll explain to you, diverticula are actually small, weird little pathological protrusions, almost like little pockmark pockets that extend out from haustra, and they are a pathology. What I was asking you on the homework was about haustra, and the haustra are the normal features that are pouches along the large intestine. As far as hormones go from the small intestine, specifically the duodenum, let's start with fatty food is eaten. It goes through the stomach, undigested, and enters the duodenum of the small intestine. First, the small intestine needs to emulsify the fat, and it needs bile to do that. So the small intestine releases cholecystokinin, and that allows bile to be released into the small intestine so it can begin to emulsify and break down the fat. In addition, it also tells the stomach to slow down so that the duodenum can have time to finish processing the fat. When protein is eaten, it arrives in the stomach where digestion begins. The pH of the stomach secretions are extremely acidic. They can be as low as a pH of 2 to 3, which is similar to battery acid. When this acidic protein mush arrives in the duodenum of the small intestine must counteract this low pH or it'll get damaged. The hormone secretin is released to stimulate Brunner's glands that are going to release bicarbonate to help buffer this acidity and bring it to more neutral levels so it won't damage the small intestine. So secretin also tells the stomach to slow down so that the protein can have time for further digestion. So secretin releases bicarbonate to buffer the pH as well as slowing gastric motility.